Hi everyone, I'm Rosa Romero, a senior research engineer and visualization researcher at Uncharted Software. In this talk, I'm going to present our ongoing work on visualizing biological graphs using a novel multi-scale approach where we combine local analysis and global context. A range of biological processes can be expressed as network phenomena. Many questions that biologists have to answer involve graph analysis, such as what biological processes or pathways lead from virus entry to protein release. Biologists start with prior domain knowledge and mental models from their experience. Then they assemble biological graphs from source literature, analyze their causal structure, and extract relevant subgraphs from them. However, the size and connectivity of these graphs make them hard to visualize and analyze in real time. Typical biological graph-based visualizations support either overviews of the entire graph with limited interactivity or have rich interactive views of focused subgraphs. However, research has shown that the multi-scale nature of biological graphs is only fully appreciated when the entire range from local to global graph structures can be inspected interactively and continuously. Our approach enables biologists to interactively explore, query, and analyze biological graphs at different scales. We utilize global and local views, hierarchical and flow-based graph layouts, multifaceted search, machine-assisted neighborhood suggestions, and drill-down operations into backing scientific knowledge. We followed a user-centered design process that involved meetings over 8 months with domain experts in systems biology, bioinformatics, and causal reasoning. We distilled 3 design goals during this process. The first design goal is to provide a scalable, interactive, real-time overview of biological graphs so biologists can gain insight into the ontological structure of the causal knowledge. The second design goal is about aiding biologists in iteratively extracting and analyzing subgraphs of interest while keeping global context in mind. The third design goal is to seamlessly link graph exploration to backing scientific literature. Biologists can therefore trace graph pathways back to source papers and then explore related knowledge to broaden their analysis. For our visualizations, we leverage datasets provided via a collaboration with a knowledge assembly system, EMA, which extracts causal assertions from thousands of scientific documents. These causal statements have mappings to biological ontologies and are assembled into a biological graph. Using such datasets, we implemented a web-based system prototype that visualizes 16 biological graphs. The main space is the graph space, which has 2 coordinated views, a global view and a local view. The global view provides an overview of each biological graph, where this graph is assembled from a large set of causal statements. Each causal statement represents regulations between biological agents such as proteins or viruses. This global view uses a hierarchical circle packing algorithm to display nested clusters of biological agents as they are organized in the background ontology. Each ontological group is rendered as a ring in blue around their children in orange. Edges are bundled together to avoid clutter. Graphs can be searched in real time using multifaceted queries. Results are displayed using contrast-based highlighting, with results foregrounded and the rest of the graph faded out. While the global view provides insight into the high-level structure, there are still sense-making challenges. For this, we coordinate this view with the local view, where the local view displays a subgraph of the causal statements that result from search queries. Here, edge color encodes different attributes such as the implied polarity of a regulation. Controls on top of an edge encode curation state, that is, if an edge has been verified by domain experts. the left-to-right flow-based graph layout encodes directionality. Selecting a local node accesses node metadata and neighborhood suggestions to aid subgraph expansion. Neighborhood suggestions are also highlighted in the global view, so biologists can visualize the global connectivity of a local node. Selecting a local edge accesses the underlying supporting evidence extracted from scientific documents. This builds trust in the analysis. We have had some initial assessments of our approach, with evaluations with domain experts in systems biology, bioinformatics, and causal reasoning. Their overall opinion was quite positive regarding the ease of interaction and the scale covered, and they believe our approach to have value. We will also continue to develop this approach and plan to do research to utilize machine learning techniques to aid search, recommendation, and ranking. Finally, we also believe that our design approach is flexible enough for similar coordinated multi-scale analysis in other graph-based domains. I would like to thank all the collaborators and contributors of this work. Thank you all for your attention.